Hello, students. Hello, students. A very good afternoon. So today we will start our session with the search for genetic material. All right. So before we start, I will launch a poll on technical check. Please let me know if my audio is clear and video is visible. And the students are from the YouTube, they can also tell through the chat box if the audio and video is visible and it's clear. All right. So let us go on with our class for now. Okay. Yeah, let us head on with our class for now. So here we have the first question. Okay. So histones are rich in alanine, glycine. Okay. First option. And second option is lysine and arginine. Third option is histidine and serine. And fourth option is cysteine and tyrosine. Okay. So I'll launch the polls again. Okay. So for an MCQ, please try to answer this within a minute. So it was from the last topic that we discussed about packing of DNA. Histones are rich in what type of amino acids? So we are almost at 45 seconds, students. So try to answer as early as possible. So I'm going to end the poll now. So let us get back to the question. So our question is, histones are rich in. So histones are positively charged proteins. Okay. So 1 second, give me. Yeah, histones are rich in alanine and glycine, lysine and arginine. So our option is lysine and arginine because they are positively charged amino acids. Since they are very rich in histones, that is what gives the histone the positive charge, which helps in coiling the DNA. Okay, so that is the answer. So let us start our session by understanding the efforts that took place in the search of genetic material. So if you remember the past, I told you that our Mendel's experiment really helped humanity to understand that there is some transforming factor, right? Factors are responsible for carrying the characteristics, but he failed to prove it through any means because of the lack of technology. So until 1928, okay, it was the preliminary efforts of Frederick Griffith. So Frederick Griffith's experiment is known as the famous transformation experiment. So in Frederick Griffith performs an experiment using two organisms. First one is mice and the second one is Streptococcus pneumonia. So Streptococcus pneumonia is a causative agent of a disease called pneumonia. So they come in two different strains. Okay, one is a smooth strain or virulent strain, we call it as. So why do we call it a smooth strain? Is because it has a polysaccharide mucus coating around it. So, and it appears smooth when we look at their colonies under the microscope, and they cause pneumonia. Okay, that is why we call it as virulent strain. Whereas the rough strain is appears rough under the microscope. Their colonies appear rough under the microscope, and they do not have any mucus coat. and they are non-virulent and they do not cause pneumonia. So what he does is, initially, first he takes a live S strain, okay, living S strain, that is, which is virulent, and he injects into the mice. The mice develops pneumonia and it dies, okay. And the second case, what he does is he takes a live R strain or the rough strain and he injects into the mice. Since living R strain, okay, is not virulent, the mice will not have any pneumonia and it survives, okay. In the third case, what he does is he heat kills S strain. So heat killing is a process of raising temperatures abruptly around 40 degrees Celsius. So when the temperatures are raised, now what happens is many proteins in the smooth strain degenerate, which is helpful for causing this virulent character. So when heat kill means the cells are alive, but it loses the ability of causing a disease. Okay. So when heat kill S strains are injected to the mice lives without causing pneumonia. Now, what he does is next he mixes the heat killed smooth strain with the rough strain and he injects into the mice. So when he injects into the mice dies. Okay. So at that point, our Griffith understood that, okay, there was some characteristics from the smooth strain that was transferred to the rough strain. That is how the rough strain turned from non-virulent to virulent, but he was not able to prove what was exactly transferred between the smooth strain and the rough strain. Okay. So today our understanding is quite elaborate. That is why we know that, whenever an unfavorable condition happens, a process called conjugation happens between this bacteria. So what happened was in smooth strain, right? In smooth strain and in rough strain had all the, proteins, all the proteins, all the proteins and factors that would help the cell to become virulent, but it did not have the gene that was causing the disease. Whereas the smooth strain, because it was heat killed, it had lost all its proteins that were supporting this process of virulency, but it had the gene intact. So because of the unfavorable condition of raising the heat, now they conjugate, okay, where this genetic material was passed down to the rough strain through conjugation, and the rough strain becomes virulent, okay. So this was Griffith's experiment. And after Griffith, okay, it took some more years to prove what Griffith's experiment was trying to say. So now three more brilliant scientists like Oswald Avery, Colin McClelland, and McLean McCarthy are the three scientists who tried to determine the biochemical nature of the transformation experiment. So during this experiment, what they do is they take these heat kill smooth strains and they treat it with several digestive enzymes like lipases, carboxylases, and then proteases, RNases, and DNases. So when they provide this particular digestive enzyme, so in the first case, when the proteases were used, correct, no? So there will be no protein. So when there is no protein, right? So now what happens is for us, when this particular smooth strain were treated with proteases, the proteins were destroyed, but was it able to cause the disease, whatever it is, like was it able to transform the rough strain? Yes, it was able to transform the rough strain. But when he uses RNases also, there was no RNA, but the DNA is intact. In both cases, the DNA was intact. That is why it was able to, transform the rough strain. Whereas in the final case, when they use DNases, the DNases that means it destroys all the genetic material in the smooth strain. So when it was mixed with the rough strain, so it did not transform the rough strain. That proved that the DNA is the genetic, DNA is the one that is actually that was transferred between the smooth strain bacteria and the rough strain bacteria. Whereas, this can give us a clear picture about what happened in the experiment. So, but it was not completely acceptable at that point. Okay. So later on, what happened to give more substantial proof, Hershey and Chase, what they did was they went even more deeper into the biochemical aspects of our macromolecules like DNA and proteins. So we know by now that the proteins have sulfur and the DNA has phosphate, right? Our DNA has free phosphate, whereas proteins have sulfur in the form of disulfide bridges. So our DNA do not have sulfur, whereas our proteins do not have phosphate, free phosphate groups. So for that experiment, they use a bacteriophage and E. coli as their experimental organisms. and they start grow and they use a medium called as radioactive phosphorus medium and radioactive sulfur medium. So radioactive phosphorus medium is the one which contains P32 isotope of phosphor, whereas the radioactive sulfur contains S35 isotope of sulfur. So now what happens? The viruses were grown in the respective medium. First, they start growing in P32 medium. Okay, so where they have radioactive DNA, because DNA has phosphorus, whereas the viruses that were grown in S35, they got radioactive protein. Now the competition was to determine whether it is DNA or whether it is DNA or proteins. And the reason why they were debating between DNA and protein is because they thought DNA is very simple to carry such vast amount of genetic information, and its variables are very less because it has only 4 nucleotides, right? Whereas when it comes to proteins, proteins are made up of 20 different types of amino acids. So that's why most of the scientists argued that it must be proteins, not the DNA, to carry the genetic material. And that is why this experiment had to be taken place. Now, what they did was, when these cells that were grown in respective mediums of S35 and P32 were infected into an E. coli cells. So when the E. coli cells were infected, immediately these cells were subjected for centrifugation. So here we can clearly see. So what happened? The protein labeled with radioactive, they were infected with to an E. coli cell. Similarly, the phase that was having a radioactive labeled DNA again infected your E. coli cells. Now what happened? So after the infection, okay, so now they do something called as blending. So when they do the blending, all the bacteriophage that was attached to the cell wall of your E. coli, they just come out. Next, what they do, after the blending, they subject this phage grown with radioactive sulfur and phage grown with radioactive phosphorus to centrifugation process. And centrifugation process, they spin the cells at 55,000 to 60,000 RPM. Okay, they spin it at 55,000 to 60,000 RPM. After they spin it or they centrifuge this particular supernatant, now they get 2 different layers. Okay, different layers in your centrifugal tube would be, one would be a pellet and other would be the supernatant. Okay, in both the cases we'll get a pellet and a supernatant. What we can observe is, whenever we centrifuge, okay, the proteins that are present in your bacteriophage, they will all be present in the supernatant region, whereas the cells and the radioactive the cellular particles of your bacteria will be in the pellet region. So that is why you can see here the radioactive sulfur can be seen only in the supernatant because it contains the proteins in case of sulfur 35. Whereas when you look at the radioactive phosphorus, the pellet region will show the radioactivity. What does it suggest? It suggests that the cells of the bacteria were radioactive. So when does the cell of a bacteria becomes radioactive? Only when a radioactive DNA is transferred from bacteria to the E. coli cells, right? So this gives a very important proof in our molecular biology, concluding that DNA is a genetic material. So it's a very important experiment and you have to remember it. So next one, coming to the properties of genetic material. So can we call proteins or RNA or any biomolecules as a genetic material? Even though RNA has certain properties of genetic material, but it is not an ideal genetic material. Okay, that is why during the course of evolution, RNA evolved to become a DNA. Okay, so let us see what are the important criteria to fulfill as a genetic material. For a genetic material, first it has the ability to self-replicate. That means whenever a new progenies of cells are produced, okay, they have to have the same same genetic information, right? So one cell divides into 2. Both the daughter cells should have the same genetic information as their of the parents. So for that to happen, the DNA should have the capacity to self-replicate. So that is the first thing. Second one is chemical and structural stability. So what is chemical stability? High reactivity or high catalytic activity of these molecules will lead to severe mutations. That is the first thing. Second thing is structural stability. So DNA is a very beautiful molecule that has high structural stability because of the presence of hydrogen bonds. That is the first cause. So it gives our DNA molecule the ability to turn, twist, and it also gives its ability to pack during the process of cell division. And next one is, even though they are structurally and chemically stable, they have to give provision for changes, constant changes in the form of mutations and recombination. So these are very essential aspects. If the genetic material do not change according to the change in the environment or change in the living conditions of an organism, then the survival capabilities of this organism would be very low. That is, you have heard about survival of fittest. The ones who do not adapt, they die, right? Next one is, it has to express as Mendelian characteristics, right? So whether it is dominance or recessive or in law of independent assortment, so they express in the form of proteins. All your characteristics that you observe in us with respect to your hair color, the skin color, the intelligence, the eye color, all of them is because the DNA is able to express in the form of characters, right? So these are some of the ideal characteristics of a genetic material, and DNA ticks all the boxes with respect to this. And next one, when it comes to the expression of your genetic material, right? So DNA, what it does is it stores the information. So the information that is stored is very vast amount of information is stored in our DNA. So if you look at humans, you can imagine the number of characteristics that we express, right? So this information is stored in DNA, but it has to be some way conveyed to the cells to have an, to form an expression. So that happens by the process of central dogma. The central dogma suggests that the DNA actually, the what do you call, the functional parts of DNA, we call it as, that in simple terms, we call it as genes, which store the information. So this information is actually converted into a messenger RNA or RNA molecule through a process called as transcription. So after a transcript is produced, this mRNA will carry the message from the nucleus to the cytoplasm, and it recruits certain machinery like ribosomes, okay, and certain enzymes, and undertakes another process called as translation, where your RNA messages are decoded to construct proteins by assembling amino acids, right? So this is very beautiful mechanism that happens in all living organisms for the genetic expression. Now, when you talk about storage of information and for the expression of information, DNA is the best genetic material for the storage of information. Whereas when it comes to RNA, it is the best, genetic material that is helpful for transformation of or transmission of information from DNA to the cellular organelles. Fine. So what about RNA? So is RNA being unstable is a problem? Yes, we agree with that. But do RNA have significance in our, in our cellular mechanisms or in our molecular biology? If you see the history of our genetics, right? The first macromolecules in the form of nucleic acids were formed were RNA molecules. RNA molecules were pretty large. And because of the structure of the RNA molecules, it makes it chemically unstable. And it provides very random and unpredictable mutations. So whenever there is a tremendous amount of mutations, now you cannot expect, your genetic materials to be stably transferred from one generation to another generation. So that is the first problem with respect to RNA. That is why our RNA was, gradually evolved to become a very structurally stable DNA molecule. Now, coming to the role of RNA, it acts as a genetic material as well as a catalytic molecule. So it catalyzes the process of, construction of proteins or formation of proteins. And you can see the RNA that is having its role in some of the important life processes like metabolism, translation, and splicing. Okay. So this is the importance of RNA, and RNA of several types. Okay, you can tell mRNA, rRNA, tRNA, okay, snRNA, etc. And we'll study their roles as we move on to translation and transcription. Okay, fine. So since we have had a discussion till now, so let us have one small questioning round. So here is your first question. In Streptococcus pneumonia, virulent form is smooth, virulent form is rough, non-virulent form is capsulated, all forms are rough. So I will launch a poll for you, so you can try to answer this. Okay. Okay. So I think the poll is visible for you, students, so you can try to answer this within a minute. So YouTube students can try to do it in the chat box. Okay, you can type it in the chat box. Okay, so we have an answer already, 2 on 2. I think we have only 2 participants, so let's go on and discuss this question. So virulent form is smooth. It's absolutely right. Okay, your answer is right. So sorry, yeah, virulent form is rough. No, it is actually wrong. Non-virulent form is capsulated. It's not capsulated, right? So all forms are rough, which is again a wrong option. So already you have chosen a right option, so no comments. But still, your first option is the correct option. Okay. Next question we have is, 1 second, I'll relaunch the poll again. Yeah, the final proof of DNA as a genetic material, okay, 1 second, came from the experiments of Hershey and Chase, Avery, McLean, and McCarthy. Okay, Griffith's experiment, and none of the above. Hershey and Chase experiment. Okay, I'll wait for 10 seconds. Okay, and then we'll continue. It's ideal to wait at least 30 seconds. No for an answer. All right. So I'll end the poll now and let us discuss this question. Okay. So the final proof that DNA is a genetic material, it came from Hershey and Chase experiment, right? So when the students who have chosen B as an option, okay, Avery, McLean, and McCarthy, first we'll discuss about this wrong options and then we'll go for the right option. See, Avery, McLean, and McCarthy, yes, they determined the biochemical aspects of the transformation experiment, but still they were not sure whether it is DNA or proteins as a genetic material. I told you. Why? Because even though they provided the proof that DNA is the one that is transferred, there was a huge debate back then suggesting that protein should have all the characteristics of a genetic material because of its number of options, right? So there are 20 different types of amino acids and protein structure is more complex compared to that of DNA. So that is why most of the scientists were arguing for Avery, McLean, and McCarthy to be, that experiments to be wrong. And finally, Hershey and Chase, they designed this experiment which gives the final answer with respect to bacteriophage and its radioactivity, right? So when we saw the pellet that was containing radioactivity, so it proved that the genetic information that was transferred from the bacteriophage to the E. coli cells was DNA, sorry. So that is why our first option is correct and the second option is wrong, whereas Griffith's experiment is about transformation and none of the above doesn't make any sense. Okay, so I hope you are satisfied with the answer, whoever has ticked B. Okay, so anyways, I hope I can move on. So if you have any doubts, you can put it in the chat box. Okay, all right, students. So let us go to the next part of our discussion. So what is central dogma of molecular biology? So it was proposed by Francis Crick. So this is one of the brilliant propositions that was made by Francis Crick as they were studying the the structure of the DNA itself. They suggested many of the things. Okay, Watson and Crick suggested many of the things, and one of the propositions that came from Crick was a very beautiful proposition. Why? Because he really told how we are expressing the characteristics and what is the role of DNA and what is the role of RNA and what is the role of proteins. So what does central dogma suggest? All the living organisms from bacteria all the way to the complex eukaryotes like humans, all of us have genetic material as DNA, and this genetic information is in the form of genes. Okay, and these genes are transcribed by a process of transcription where our enzymes and proteins, they synthesize your nucleus to the cytoplasm, and through the translation machinery, now what happens is your RNA is converted into proteins, okay, by translation. So this is called as central dogma of life. Okay, guys, I have to confirm one more time. Is my voice and audio clear? Please let me know. Are you able to listen continuously? Please let me know, because I'm getting a notification of network problem. Just make a quick. Since there was a problem with network, I just want to know if my audio is continuous and video is visible. Just let me know so that we can continue on faster. Okay. Hi students, can you see or hear me? Can you let me know? Okay, fine. I'll stop sharing the technical check. I will continue on with the class. I hope that's okay. If you have any problem with technicalities, please let me know in the chat box. Okay, if you hear me out. Okay, so let's see. So we talked about central dogma of life, but in some cases, what happens is in most of the organisms, all of the organisms who have DNA as a genetic material, this is the flow of life. DNA to RNA and RNA to protein. But you can see some of the viruses, they have RNA as their genetic information, primary genetic information. But when they come to the infectious stage, for example, your HIV virus, what happens is the RNA is converted back to DNA through a process called as reverse transcription. And through DNA, again, the RNA molecules are synthesized and the new viral particles are produced. So however it has to start from DNA, okay? So you have to remember the concept of reverse transcription. Reverse transcription suggests that the RNA molecule, RNA is converted to DNA, and this process is called as reverse transcription, and it is mostly seen in the viruses which contains RNA, okay? So let us go to the next topic, that is DNA replication. So when it comes to DNA replication, this is one of the milestones in understanding how our DNA is able to make its copies. So when it comes to that, if you remember the cell cycle and cell division, so we know that our cell cycle can be divided into several phases, right? For example, if I just write a random cell cycle, so we have G1, then you have S phase, and then you have G2, and then you have M phase, and then you have G0 phase, right? So this is how the cell cycle happens. In the S phase, what happens is the replication of DNA takes place. So this is where the replication happens. So to prove what type of replication or how the DNA is making its copies, okay, there were several hypotheses, or there were several propositions that was put on. Okay, today we accept widely the semi-conservative model of replication that was suggested by Watson and Crick, and later it was proved by Michelson and Stehel's experiment. Okay, so before we could prove that semi-conservative model is the ideal model of replication in all the organisms, there were other modes like conservative and disruptive mode of replications. I think we will discuss this in the revision class. So when it comes to semi-conservative mode of replication, it says that semi means half, no? So semi means what? Half is equals to half, correct? Or 50%. Conserved means what? Saved. So whenever we talk about parental strands, so our DNA is a double-stranded, right? It is double-stranded helical structure, right? So when we talk about two strands, let us say that two strands are both parental strands. So when these parental strands replicate, now what happens in the newly formed daughter DNA, there will be one parent strand, okay, like this, and then a newly synthesized daughter strand. So for every new DNA that is formed, there is 50% of parental strand and there is 50% of daughter strand. So that is why we call this model as semi-conservative mode of replication. So let us see the experimentation that proved, okay, the semi-conservative mode of replication. Okay, so now what happens? This experiment was conducted by Michelson and Stehel. So for this experiment, they take this organism. The organism for this experiment is E. coli. So what they do is they grow this E. coli in two different medium. In the first medium, what they do is they grow this E. coli in nitrogen containing heavy isotope, that is ammonium chloride they take. And in ammonium chloride, the nitrogen is of heavy isotope, okay, 15. Whereas what they do after they start growing E. coli in heavy isotope for several generations, they make sure that the entire DNA, right? The entire DNA is incorporated by N15 isotope. If you keep on growing for several generations, now the DNA becomes N15. Now, how do I say this? You become what you eat. No, whatever you get in your surroundings, it gets into your body, and your body metabolizes and uses the same products that you have put in your body to construct proteins, DNA or RNA, etc. It is the same case in all the organisms. Okay, so when we grow E. coli for in heavy isotope for several generations, after some time the entire DNA will be made up of heavy isotope of DNA only. So once we have confirmed that all the entire DNA becomes heavier isotope, next what they do, they prepare one more medium containing normal isotope, that is N14 isotope, or we can also call it as lighter isotope medium. And then they introduce these E. coli cells from N15 medium to N14 medium. Okay. Now time is very essential. Why time is very essential? For every 40 minutes, our E. coli cells, okay, they divide. So since they divide for every 40 minutes, that means in the mid of their cell division, at exactly at 20 minutes, now the replication should have happened. Correct, no people? The replication should have happened. So what does it suggest for us? So what they do is they divide the timeline for every 20 minutes. Okay, 0, you can see here 0, 20, 40, 60, and 80. They keep on dividing the timeline like for every 0, 20, 40, 60, 80. First case. So what happened? We took the N15 cells and we introduced into the N14 cells. Immediately after we introduce the cell into N14 medium, now what we do, we perform density gradient centrifugation, where we use cesium chloride as the gradient, okay, density gradient. So cesium chloride is used because it helps in the separation of the DNA at low RPMs. So once we perform the density gradient centrifugation with cesium chloride, at the 0th minute, what happens? Since the cells are freshly introduced from N15 to N14, so all the DNA will be of what? N15 nature, isn't it? So that is why we get what? N15 band. We'll get a clear N15 band. Okay. So next what happens is, after 20 minutes, the replication cycle would have completed. So after the replication cycle would have completed, now what happens? Our DNA will form a new band. Okay. For example, 20 minutes happened, right? For example, you have your, let us say, N15. After 20 minutes, what happens? We have N15, N14. Correct? No? And again, we have N15 and we have N14. Sorry, I've written on the reading. Okay. So, but you understood, no? We have N15, N14, N15, N14. Correct? No, that is what is semi-conservative. So when that happens, we have a new band which is of 100% made up of, which is quite lighter, I can say. So this band is in between. Okay. So it contains N15 and N14 band, we call it as, that is intermediate band. Understood? No? See what happened? I will just explain this once more so that it will be easy. Okay. So we have N15 and we have N15 as the parent strands, correct? So when they replicated, we get two DNAs. So one strand will have N15, N14, and again it will be N15, N14. Clear? So that is why when we centrifuge, it will form a 100% new band, intermediate band we call it as, between N15 and N14. Okay. Next, what happens after 20 minutes? Again, at the 40th minute, when we do centrifugation, again, now the cell division has completed. Now what will happen? There will be a second cycle, isn't it? Now this, let me erase this and I will try to explain it again. See what we had N15, N14, correct? So this N15, N14, again, when it replicates, it will form N15, N14, and again we have N14, N14. Same way, we have second one. No, this is for one DNA. We have another DNA, right? So that will form what? N15, sorry, N15, N14, and then again we have N14 and N14, correct? No. So if you see this, it should form two bands. So since we have equal amount of N15 and N14, so it will form one band of N15 and another band of intermediate band, that is N14, 50%, 50%. Next, after if you do for the 60-minute cycle, you will have 75% of the band will be of N14, and then you have 25% of the intermediate band, that is N15 and N14. And as you go further, now what happens? 88% and 12%. It will keep on reducing it by half. You can see here 100 to 50, 50 to 75, 75 to 88. Or if you see in the reverse case, it will be 100 to 50, 50 to 25, 25 to 12%. So this exact reduction in the percentage of your heavier isotope of your DNA suggests that the DNA has to replicate through semi-conservative mode. In any other cases like disruptive or conservative mode, your DNA cannot have this perfect mathematical distribution of 50%. So that proved that the DNA replicates through semi-conservative mode. Okay, so you can see here clearly the further explanation of your Michelson and Stahel's experiment. So fine. Do you have any doubts, students, with respect to what we studied about Michelson and Stahel's experiment? Okay. So please put it in the chat box or send a reaction. So it would be very nice so that we can move on to topics. Okay, give me a second. I'll just check on with YouTube. No problem. So YouTube students, you can just put it in the chat box if you have any doubts. Okay, we can sort it out. Fine. So let us get back and talk about DNA replication. So I think the time would be sufficient. Let's try. So DNA replication, as I said, it replicates through semi-conservative mode, and let us understand the mechanism of DNA replication. So mechanism of DNA replication is a little bit complex, but it is all right, because if we understand step by step and map the process, it's very easy for us to understand. Okay. So first, let me change the ink color to white. Fine. So now what happens? Double strand, our DNA is double stranded, right? So the double stranded DNA, initially what happens? It has to be unwound. That means it has to be split. So before anything can happen with replication, the first obligation for replication to even happen is the presence of a very conserved sequence called as ORI site. Okay? ORI site is also called as origin of replication site. So this origin of replication site is present in all the living organisms. Now what happens is this ORI site, the number of ORI site actually varies. If you see in prokaryotic organisms, okay, if I write prokaryotes, there is only one ORI site. If you see eukaryotic organisms, okay, like humans, we have hundreds of thousands of ORI sites, okay. So I'll read it as n number of ORI sites are present in our eukaryotic cells. So that is a primary difference between prokaryotes and eukaryotes, very important difference. So once we have an ORI site, what is ORI site? ORI site is the region which is conserved. So what is the meaning of conserved? Conserved means for several millions of generations, these sequences are preserved very well from one generation to another generation, and they are not lost. So these conserved sequences are very essential for recognizing the site to begin the replication process. Now, once we have an ORI site, the ORI site is usually very rich in adenine and thymine, nucleotides. The reason why they're very rich in adenine and thymine is because it is very easy to break these double bonds rather than triple bonds, which is present in GC rich region, correct? No. So once this ORI site is recognized, now what happens? There is a special enzyme called as helicase. Okay. Now this helicase comes and recognizes this ORI site and binds to the ORI region. Once it binds to the ORI region, now what happens is helicase will hydrolyze the bonds between your adenine and thymine or whatever the base pairs that are present in your ORI site. Once it hydrolyzes the bond, it splits your DNA into independent strands or unzips the DNA. Once the DNA is unzipped, okay, now our DNA is quite, it cannot remain, split. It will immediately join, okay, the moment it is split. So now what happens? Your helicase that is bound to the ORI site will start moving further. As it moves further, now what happens? Your unzipped DNA should not zip back, no? So for that, what happens? We have special proteins called as single strand binding proteins, or SSBP we call it as. Here it is. So single strand binding proteins, they go and bind to the nucleotides and they temporarily stabilize them, preventing them from forming the bonds again with their, with their natural strands or with their complementary strands. Now what happens once we have our SSBPs stabilizing our both single stranded DNA? Now we have our very important, okay, very important enzymes. Those are called as DNA polymerase. Okay, these are called as DNA polymerase. I'll write it as DNA pol. So the DNA pol is of three types. Okay, DNA pol 1, DNA pol 2, and then we have DNA pol 3. All of them have very important functions. Okay, let us discuss one by one. So before we head on to what is role of individual DNA polymerase, now DNA polymerase has a problem. What is the problem? Always DNA polymerase can synthesize a new DNA strand in 5' to 3' direction only. Now, why is the question? Why does it synthesize only in 5' to 3' direction? The answer for this question is, if you look at your sugar group that is present in your DNA, it has a 3' OH group, isn't it? At the third position, there is a free OH group. So this 3' OH group is essential initially. Okay, first there should be a free 3' OH group. Only then it can use this free 3' OH group and start making connections with the new nucleotides that is coming. That is why our DNA polymerase cannot start the process of polymerization de novo. De novo means on its own. So it takes the help of other factors. We'll discuss about it. So this is the first nature of DNA polymerase. Coming to the function of DNA polymerase, the first one is DNA polymerase 3. DNA polymerase 3 is very important because it continuously synthesizes DNA. Okay, it does what? DNA synthesis from 5'. Okay, it does that. Next one, what happens to DNA polymerase 1? DNA polymerase 1, what it does is, in lagging strand, okay, we'll talk about it. In lagging strand, okay, it will produce DNA RNA primer. Okay, this is the first function. And the second function is, it will remove RNA, okay, RNA primers. It will remove RNA primers. Two functions. First, it will help to build DNA RNA fragments. Next one, what it will do, after the construction of DNA RNA fragments, it will remove the remaining RNA that is present in the stretch of DNA. I will discuss with respect to it in detail. So when it comes to DNA polymerase 2, it will do very important function, that is proofreading, or it will try to recognize if there are any process of replication. So let us come to the beginning of replication. So what happens? Once your strands are unwound, okay, we have two different strands, isn't it? One second, give me a second. I'll just erase it. Okay. Okay, we have just 5 minutes. I'll try to just talk about a little bit about DNA, replication, and then we can continue in the next class. So let's see what we can try to finish. So let us say, so we have DNA pol 3. Okay, pol 3. Now what happens? Our DNA is split into two strands. Let us say that one strand is going from 5' to 3' direction, and another strand we should go from 3' to 5' direction, isn't it? I told you the DNA replication always happens from what direction? 5' to 3', correct? No, so this should be our continuous strand, we can call it as. Now, before even our DNA polymerase can start the polymerization reaction, it takes the help of RNA primase. Okay. So what does RNA primase do? RNA primase will lay down a short primer. Okay, in what direction? 5 prime to 3 prime direction. So when it lies a short RNA primer from 5 prime to 3 prime direction, let us call it as RNA. Now what happens? What is the benefit for the DNA polymerase 3? It has a free, where is it? Okay, where's my ink? Okay, sorry. Yeah. So we have a 5 OH. Okay. Now what happens? A polymerase will start synthesizing the new DNA strand continuously. Okay. I'll write it as continuous. Okay. Or leading in 5 prime to 3 prime direction. And this keeps on happening. Okay. Whereas when it comes to other opposite strand, now what is happening? Your DNA polymerase can only synthesize from 5 prime to 3 prime direction, isn't it? Now, if you see the unwinding of your strand, if you see that helicase is here and helicase is moving in this direction, if you take, okay, so let us say what is happening here. Here, our DNA should be synthesized in opposite direction, isn't it? So if the DNA has to be synthesized in opposite direction, that means periodically our DNA polymerase would require, 3 prime OH again and again. Okay, sorry, again and again they would require 3 prime OH. For that, what happens? Give me a second. Yeah. So for that, what happens? An RNA primer comes and then it will lay short RNA primers, okay, periodically, okay, here and there. Now let us call it as, I'll just write it as RRR so that you understand. This is RNA primer that is being laid down, periodically, like tiles or steps. Next, what it does is, 1 second, I will change the ink again. Okay, I'll put it as white. Okay. Now what it does is DNA pol 1, okay, DNA polymerase 1, it will use these RNA primers and it will construct, okay, DNA RNA primers, correct? No, we have DNA as well as RNA. I'll write it as DDD. Okay, DNA RNA primer it will construct. Once this DNA RNA primer is constructed, again the DNA polymerase 1, what it will do? It will do something called as exonuclease activity. Exonuclease activity means our DNA polymerase will have the ability to cut the RNAs from end to end. Okay, one end and another end, it will cut it off. So once it is cut off, now what happens? These sites which are blue will become empty, no? So these empty sites is again elongated by DNA polymerase 3. Okay, DNA polymerase 3 will come and elongate this DNA primer. So when it elongates, if you see in the lagging strand, we call it as lagging. Okay, lagging strand. If you see in a lagging strand, you have DNA fragments, something like this. Okay, as fragments. So these are called as Okazaki fragments. So these, it was discovered by a Japanese scientist called as Okazaki. Now what happens? The ligase, okay, DNA ligase is another enzyme. So the DNA ligase will come, it will use the 3' OH and the 5' phosphate group, and it will create the bonds. Okay, what bond? Phosphodiester bond between the nucleotides, and it will make it continuous DNA. So these are the activities that is happening in leading and lagging strand. Alright, people. So now what happens? The DNA polymerase 2 will do the proofreading, and as our helicase is moving forward like this, our DNA is coiled, no? So since it is keep on unwinding in the ahead of your DNA, there is a possibility of creating something called as torsional stress. Torsional stress. So when a torsional, like just like you take 2 ropes and you unzip suddenly, what will happen? It will be super coiling will happen. No. So that means your rope will become knotted at the other end. So this is called a super coiling. So when that happens, there is a probability of your DNA getting nicked or broken. So to avoid this torsional stress, there is one more important protein called as gyrase or topoisomerase. Okay, so gyrase or topoisomerase, what they'll do, they will move before the helicase, relieving this torsional stress, so that our helicase can leisurely open, okay, or without any worries, it can open our DNA strands into two strands for the process of replication to take place. So if you see the pace of replication, both your leading and lagging strands almost have the same pace or same speed. Because of the complexity of the lagging strand, we call it as lagging strand. But if you see the speed of reaction, it is almost same. So this is a summary of DNA replication, and we will continue in the next class and summarize about it. Okay, by now I will end the class. Okay, we'll see in the next class. Thank you, students. Okay, so you can leave now.